Okay, I think we can get started now. So good afternoon, everyone. Welcome to the TransPot Collaborative Symposium. The subject is Integrated Prostate Cancer Research across the European Union. My name's Katie Wright and I'm the Project Manager of the TransPot project based at the University of Glasgow. Hopefully you've all seen a copy of the agenda for today's symposium. So we have several speakers, including early stage researchers from the TransPot project, as well as guest speakers Bruno Costa-Silva and Teijo Pellinen. So special thanks to our guest speakers for agreeing to join us today. We have quite a few people on the call, which is great. Can I ask, if you're not presenting or chairing a session, to please keep your microphone muted and video turned off. We'll be having a short Q&A session at the end of each presentation. So if you have any questions you'd like to ask, can I ask that you add them to the chat function? That's the speech bubble at the bottom of your screen. And the session chair will gather all questions and ask those to the speaker at the end of each session. So any questions can just go in that chat function. Our symposium will be recorded for dissemination purposes. And our first speaker is Professor Hing Leung from the University of Glasgow, who is our TransPot coordinator. So if I can hand over to you, Hing, for the first presentation.
Thank you. Good afternoon. So it's a really great pleasure to introduce the TransPot project to everyone. First of all, I want to say thank you very much for joining us for this collaborative symposium organized by the TransPot network. I should say that a lot of the work actually were done by Katie, Craig Robson, and the training committee. So I'm really just the messenger boy here, giving you a summary of the overview of the project. It is a training network. We're particularly keen to enrich the training experience for our early stage researchers. And we've engaged them with giving talks and also introducing our speakers today. So I want everyone to support them with questions at the end of each talk to enrich the training experience for everyone. We're very keen to, I'm having slide, okay, yes. We're very keen as a network to consolidate and develop research collaborative opportunities. And for those who are not familiar with the TransPot project, you will see the web page on each of the slides that I'm presenting. At this web page, you'll see details of the TransPot projects. On the right side, you'll be able to find information about particular projects and also how to contact individual research teams within the network. One of the key aspects within the innovative training network is actually about secondment as a tool for enriching the training opportunity and also to develop research collaboration across the network. This highlights the particular opportunity we plan out at the start of the project. I should say that we're now 1 year to the end of the project, and we have taken the opportunity to refine the secondment arrangements depending on the student's project and also the training needs. So that's very important and very pleased that we have been able to do that. As a network, we are keen to develop new and also adjunct biomarkers for castration-resistant prostate cancer. Here's a very simplified cartoon to highlight that despite hormone manipulation or androgen deprivation therapy, the cancer marker here, shown as PSA, is almost inevitable to rise despite favorable treatment response judged by the PSA. So there is a very urgent need for better treatment to target the androgen receptor pathways and also better marker to report to us which patients are at particular risk of relapse. As a clinicians, I'm particularly keen to translate and progress novel findings from the laboratory to the clinical evaluations. Within the TransPot network, we have three pillars or three main tools. First is biology, second is informatics, third is clinical resource. And through the application of all three resources, we are seeking to discover new treatments and target and biomarkers, and also then validate these novel findings. Now I'm going to highlight individual projects briefly for you, so you can get a sense of the program, and you can find out more through discussions. The first project is led by Raphael in Glasgow. His interest is to study SLFN5 as a key molecule in castration-resistant prostate cancer. SLFN5 was initially found as a key protein from a number of prostate autograft studies done by Mark Sauji and one of the group leaders at the Peterson Institute, Sarah Sullivan, using proteomic analysis. Raphael has shown data using various model systems highlighting SLFN5 its role as crucial for in vivo CRPC growth through its effect on cancer metabolism. So this is something that he's pursuing at the moment, and hopefully finalize this data before he leaves. The next project is led by Parvien under the supervision of Craig Robson and Rakesh here at the University of Newcastle. Parvien has applied a very novel human-induced pluripotent stem cells to regenerate prostate organoids. And through that, he is asking the question, what are the key genes and regulators that control the developing prostate epithelium through a range of transcriptomic analysis? And through that, one would hope that their role can also be interrogated in the development of prostate cancer. Umbrint based in Belfast with Ian applies novel methods with ChIP-seq and ATAC-seq to ask the question, which are the regulators that is influenced by androgen receptor or castration-resistant prostate cancer. Afshan, working in Turku under the supervision of Perku and Matthias, has an interest in FGF receptor, particularly FGF receptor L1, and she has extensively collaborated with the groups in Belfast to seek the interaction between androgen receptor and how FGF receptor L1 drives prostate cancer progressions and metastasis. Annelies, you hear from herself this afternoon. She, under the supervision of WISC in Rotterdam, applies novel PDX or patient-derived xenografts and organoids in the purpose of identifying novel targets and hopefully developing new therapeutic agents. Marouane, based in Numaris under the supervision of Eric, is also talking this afternoon, and he will explain to us how he applied NMR spectroscopy to identify novel urinary-based marker panels for prostate cancer and also resistant disease. Anna, based in Athens, working with Tonia, applies cutting-edge proteomic methodologies to study clinical specimens to seek the application of system biology analysis to understand which are the altered pathways that are associated with treatment resistance. Mina, based in Tampere University under the supervision of Tapio, has really the advantage of applying highly novel data from unique patient cohorts in the study of long coding RNA in their role in prostate cancer metastasis. And we look forward to her talk as well. Mario, based in GenomeScan, under the supervision of Ines and Bart, has got a hypothesis that Regulant is critical in prostate cancer and in treatment resistance. And he will show us some of his network analysis data that suggests this is valid in prostate cancer. Lastly, CAFO, based in ALMEC, one of our industry partners, supervised by Richard Kennedy and Nuna McCabe, has been applying the proprietary signatures in forming prostate cancer subclasses that may have a prognostic implication. So here I have very briefly highlighted the various projects. I want to thank all my collaborators and the enthusiastic input from our ESRs, and I would be very happy to take any questions if time permits.
Thank you, Hing. It doesn't look like we've had any questions yet. So if there aren't any questions, we can move on to the first session, which will be chaired by Saida Afshan. So if I can hand over to you, Afshan, for the first session.
Yeah. Good afternoon, everyone. Hope everyone is keeping well. My name is Saida Afshan. I'm a PhD student in TransPot at University of Turku. I'm pleased to welcome our first speaker for today's symposium, Dr. Bruno Costa-Silva. Dr. Bruno is a principal investigator in System Oncology Group in Champalimaud Research Foundation in Lisbon. The general interest of his group is to understand how non-tumor cells contribute to tumor initiation, growth, and metastasis. Today, he will be sharing with us his expert opinion on extracellular vesicles and their role in metastasis and therapy response. With this, we welcome you, Dr. Costa-Silva.
Hi, so good afternoon, everybody. Are you listening to me? Can you listen to me?
Yes, we can hear you.
Okay.
Thank you.
Yes, we can hear
That's
you.
fine. So first, I'd like to thank for the invitation to participate of the meeting. And I mean, as you're going to see today, like most of my work is not exactly in prostate cancer, but I think that several of the aspects that we pursue in our research can have important value to be explored in prostate cancer. So I always like to start. Let me see here. Here. So I always like to start like my main motivation to join this research area. And this comes from this paper in 2005. Something that, firstly, like for me at this time was still novel, the idea of tumor-associated cells. Like, of course, you look to a tumoral lesion, you won't have only tumor cells. These cells are mixed with leukocytes, with other cells like fibroblasts, endothelial cells. So I mean, tumor lesions are more complex than only tumor cells, of course. But these cells are not only there sitting next to the tumor, but they play a very important role in interacting with the tumors, providing, for example, growth factor, also supporting the remodeling of the microenvironment, like extracellular matrix. I mean, blood vessels, of course, they can provide both nutrients working as a conduit, but also they can release growth factors. So, I mean, this idea of the tumor microenvironment at this point for me was enough interesting. But something that it became even more interesting in this work was the idea of pre-metastatic niche. So the idea that, I mean, of course, like one of the key questions if you want to avoid death by cancer is to understand how metastasis happen. And of course, not only understanding, but if possible to predict whether and where metastasis will happen. And these guys, what they showed is like a lot of these modifications in the microenvironment, they were working in the model of melanoma that were metastasizing to the lung. And what they saw is that a lot of these changes that I described, like in tumor-associated cells, were happening like much before the first metastatic cell arrived in the lung and populated the organ. So, and they were showing by several angles how these modifications were key for those tumors to be able to metastasize to these specific organs. Although this paper was very interesting and attracted my attention and of other people that I ended up working later, what they didn't have at this point was a very clear understanding of the mediators. So how come a melanoma that were sitting in the skin of the mice or the patients could be releasing whatever, like we didn't know what was the mediators, but something that probably could be traveling through the blood circulation and acting, for example, in the bone marrow, in recruiting many of those cells to then go to these pre-metastatic microenvironments and then set up these proteomorigenic niches. We didn't know, they didn't know what was the mediator. Something that I was fortunate. It was like 5 years later, I had the opportunity to join this group, start working there. And something that I was interested and they were interested as a potential mediator were extracellular vesicles. Like, I mean, you guys may probably heard a lot about them before, but just to give you an idea, they are usually tiny. They are around from 30 nanometers to 1 micrometers, like the most prevalent types, exosomes and microvesicles. Something that's very interesting about them is that they carry every basic biomolecule that you find in a cell, like nucleic acids, proteins. They are abilipidic structures. They carry also lipids. There are even more recent reports telling they can carry even metabolites. And not only carry, like this part of the carry is interesting because if you learn how to catch them in the circulation, in biofluids, can be like in blood, cerebrospinal fluid, urine, saliva, you have an opportunity to get, by a non-invasive method, access to the molecular compositions of cells that are present in this organism. And something that now more for the biological interest, something that's also interesting is that a lot of these molecules in here can, by being packed in these extracellular vesicles, they can be transferred to other cells. Like, and when transferred to other cells, they can actually affect a lot the biology of these recipient cells. So they could be also playing a role in cell-cell communication. As we are working in cancer and with a translational interest, these two aspects were very interesting for us. Number one, the biology that we wanted to understand, but number two, also whether we could use the information that we learn in here in the biology, whether we could use this to go back to the patient samples and try to use it to diagnose, to stratify patients with risk of developing metastasis. And actually, that's what we did. This paper we published in 2012. We saw that extracellular vesicles coming from melanoma, the very same model I was talking before, they were sufficient when, for example, when you get these vesicles and inject in a naive mice, an animal without tumor, we could see that all these features, recruitment of bone marrow derived cells, the cells populating the lungs, we saw vascular permeability in the lungs as well. So all these processes could be induced by those extracellular vesicles coming from the tumor, giving us a very strong suggestion that they could be the mediators of pre-metastatic niche setup. And not only that, as I told you, like we had by doing that, we also have an opportunity to get a closer look at the molecular composition of those vesicles and ask whether we could use these molecules that play a role in pre-metastatic niche setup, whether we can use this to diagnose patients, for example. And here, what we did, like, I mean, this is in stage 4 melanoma patients, and this is very, very simple. Like, you isolate the vesicles, you look for the protein content in the isolate that you have, and here it's pretty much dividing patients that had vesicles with more or less than 50 micrograms of proteins. And the patients with more protein content were exactly those with the poorer survival when compared to those with less content. This is suggesting us that, I mean, the more information you have in those vesicles, theoretically, the higher the risk, and probably mediated by the capability of those vesicles to transfer information from one cell to another, of having a worse progression. And here was also interesting because this molecule, TRP2, we saw as one of the key molecules in mice to set up these pre-metastatic niches. Now looking for stage 3 melanoma patients, we could see that those patients with higher levels of this molecule that was related with pre-metastatic niche setup were exactly the patients with progression of disease after within the 5 years of follow-up. So again, here was to talk about the survivor, and here to talk about progression to development of distant metastasis. So So this was in mostly studying the lung microenvironment. Something that I also did was to take a look in tumor models that had a preferential metastasis for liver. Here we work with pancreatic cancer. We saw that this pancreatic cancer-derived vesicles, they could specifically bind to the liver. I won't be going into many details in here. You can go back to the paper if you want to get more information, but these vesicles played a very important role in modifying the activation status of resident macrophages in the organ. And then by modifying this inflammatory status of those cells, you had a chain of reactions that was involving remodeling of extracellular matrix, arrival of bone marrow derived cells, like immune cells, and those cells are finally providing the support for the metastatic cells to arrive in the liver. Something that we found in this paper was that this protein here, macrophage migration inhibitory factor, it was playing a key role in this process. So, I mean, what we did was pretty much to go back to the patient samples and ask what does the measurement of this protein in the vesicles from the patients tell us anything about the progression. So this is the levels in health donors. These are the levels in stage 4 pancreatic cancer patients. And here are patients that at the presentation, they didn't have imagiological evidences of metastasis. These patients, they never progressed to metastasis, and these patients progressed to metastasis within 5 years of follow-up. And you can see here that the levels of macrophage migration inhibitory factor, this molecule in here, are higher in those patients. So the idea is that we could more or less predict which patients were going to progress to liver metastasis by understanding the biology of this process. But now about prostate cancer. I mean, as I told you, I don't work with that, but I mean, there are a lot of very interesting questions that still have to be answered. And probably one of the ways to approach that is by studying extracellular vesicles. Here's just to do a quick review of what has been done so far. As I told you, something cool about extracellular vesicles is that, again, you can get a glimpse at the molecular composition of every cell of the body, actually, including tumor cells. I mean, every cell in the body produces extracellular vesicles, but you can look for something specific coming from a tumor, like in this case, slice variant 7 of AR. In this case, they were looking for the mRNA, and ask, like, and this is like a blood exam. These guys, they measure this molecule here in extracellular vesicles in plasma. So here is just to tell you, like, I mean, they could get a very interesting glimpse at the presence or absence, or at least looking at the levels of these slice variant 7 in the vesicles, and this could be giving an idea of what was the scenario in the tumors of those patients. And you can see here that those patients that they didn't find copies of the ARV7 in the vesicles were exactly the patients with a better survival when compared to those that had this slice variant 7 in there. This is progression-free survival. This is overall survival. And something that they went even deeper, I think it's very interesting. What they found is that not only by having or not having copies of this slice variant, is actually the more copies they found in the vesicles, the worse was the prognosis of the patient. So they could get even into this detail of substratifying the patients that had this variant in the vesicles. Here's very quickly, like, I mean, the same thing that they did in here in plasma was pretty much done with urine. So, I mean, even less invasive, like this is even simpler than the study before. So you can collect urine from the patient, and you can easily isolate vesicles from the urine, and then study whichever molecule you think is connected with a biological aspect of interest. And these guys were looking again to the slice variant 7 of AR, and once again they found that patients, the castration-resistant patients, were exactly those with higher levels of the slice variant when compared to the other groups. Here's another example. This is a correlational study. Like, the main question that they had were they had some in vitro models of prostate cancer, and they wanted to ask, the question was, can we find a molecular difference, in this case proteins, in the vesicles from resistant or sensitive prostate cancer cells, I mean sensitive to the docetaxel, and can we use this to predict or to measure, to early measure the resistance of the tumor of those patients to this drug. So what they found was like they found some proteins that were specific for resistant cells-derived exosomes or for sensitive cells-derived exosomes. They got especially interested in these MDR1, MDR3. These proteins are related to the, they are pumps pretty much. They've been described before in cases of chemoresistance by removing drugs from the cells. And what they found in here is like, I mean, this is in vitro, so just confirming what they were seeing here, but this is, I think, more interesting. They had not many patients, but had 6 patients, 3 patients sensitive to docetaxel, 3 patients sensitive, resistance to docetaxel, and they found the levels of these 2 proteins higher in the resistant ones when compared to the sensitive. So this could be theoretically applied to early measure, to access, I mean, even before treating the patient, theoretically, you could measure that and see the chances of this patient to respond to this treatment. And this is, in this case, again, was performed by measuring extracellular vesicles in plasma of the patients. Now, in addition to the 2 correlations and to the classic liquid biopsy, I think something that is also important to highlight is that there are already some reports, they are mostly recent, talking about the biology, like what can prostate cancer cells-derived extracellular vesicles do to cells that are usually key for example, metastasis, right? So bone metastasis. So this is just to give you some examples of recent works that, and they are pretty much telling the same thing, like these vesicles coming from a prostate cancer cell and acting on the maturity or even in the presence or activation of osteoblasts. And they are what they saw was that they are all working in the direction of performing bone remodeling that could be theoretically ideal for, I mean, to receive the metastatic cells in the bone. I mean, so there are some interesting reports on that. I think it was nice to try to bring it up today. But more than that, like in addition to this effect in the osteoblasts, there are also some interesting reports on effects of prostate cancer-derived extracellular vesicles in other cells, like, for example, myeloid cells, macrophages, for example, the effect of these extracellular vesicles in promoting immunomodulatory phenotypes in those cells, potentially protumorigenic. Also effects in endothelial cells, like there are interesting reports in the promotion of proliferation and formation of new blood vessels stimulated by those prostate cancer-derived exosomes. They could be also playing a role in the tumor microenvironment, either the primary and the metastatic. Also effects in stromal cells like fibroblasts, for example, activation of these fibroblasts and having these fibroblasts remodeling the extracellular matrix, producing extracellular matrix molecules, and also some growth factors. So again, there are several, I think, more and more evidences that, I mean, as in other tumor types, in prostate cancer, you probably see these vesicles playing a lot of important roles that could be linked with tumor progression or including metastasis. This is a paper that I found also was quite recent. I think was at the end of last year. What they found was like one of the first evidences of pre-metastatic bone in cases of prostate cancer. So modifications in the bone that would be happening much before the formation of metastasis. I mean, and this is very interesting, of course, because if you find the molecules that are mediating that, you can predict the risk of bone metastasis. And also, if you learn which are the molecular modifications that are happening in the bone, this could be also an opportunity for you to detect the risk, like the modifications in the bone that would be linked with metastasis. And of course, the last step would be, if you understand this molecularly enough, you could in potential develop drugs to try to prevent or revert modifications in bone that could be linked with prostate cancer bone metastasis. This, what they did in here was to pre-expose the animal to prostate cancer-derived extracellular vesicles. This is animals treated with PBS. These are animals treated with extracellular vesicles. What they did is, after exposing the animals to these vesicles for some time, they challenged the animal with an intracardiac injection with prostate cancer cells. And what they saw is like this, they saw more number and more burden of metastasis in those animals, including the bones. And this is also very interesting. Here they did the same thing, but instead of getting extracellular vesicles from prostate cancer cells in vitro, they got plasma from healthy patients and plasma from prostate cancer patients with bone metastasis already. And they saw the same thing. Vesicles coming from the plasma of those patients, they were capable to somehow modify the bone microenvironment that could finally receive more metastasis than those animals that were never pre-exposed to those extracellular vesicles. And this paper is very interesting. They went into many details on the mechanism of that, but something that was interesting was like they found that bone marrow-derived stem cells, they were producing more CXCL12, and the hypothesis they brought up was that the CXCL12 was actually acting in the CXCR4 in the prostate cancer and making these cells increasing the survival, increasing the proliferation of those cells in the bone microenvironment. So that was the proposed mechanism. And of course, this could be, I mean, potential targets if they really verify this process in other models. Something that we are doing also in other models, but I think that it's very, based on what I just showed you, it could be feasible to test in the model of prostate cancer, is like, so far we have ways to tell if a tumor is more or less prone to form metastasis, like in the case of pancreatic cancer, melanoma, breast cancer, what we work more in the lab. But something that we cannot do is to measure by a non-invasive method whether a premetastatic niche or protomorigenic microenvironment is formed or is being formed in an organ. Of course, I don't think it's going to be very ethical or even feasible to perform a biopsy of the bone or the liver or the lungs from a healthy patient or from a patient without evidences of metastasis, based only on the study of one or two molecules coming from tumor exosomes. So we probably need another hint to tell whether this patient is already developing a protomorigenic prometastatic microenvironment. And I think this is very important because at least something we are pursuing in the group is to, if possible, one day try to revert these modifications in the microenvironment and try to prevent metastasis. This is our long-term goal. So for us to get to this point, we have to have a method to detect these modifications in there. And well, I told you that extracellular vesicles are not released only by tumors, they're released by every cell in the body. So we could use the very same strategy, isolating extracellular vesicles from biofluids, to do that. This is the idea. And just to tell you that is not totally, absurd. There are other people doing these sort of studies in non-oncologic settings. This is just to give you an example. People are using extracellular vesicles in plasma to substratify pathologic status of the liver, for example, like hepatocellular carcinoma or hepatitis B infected liver, or cholangiocarcinoma, for example. And for doing that, they're using, like, for example, microRNAs in extracellular vesicles. So again, it's feasible not only for tumors. In order to do that, I mean, we in the lab we still work a lot with the liver, like, because of our interest in pancreatic and colorectal cancer. So as I told you, we can induce these microenvironments by giving sequential doses of tumor-derived vesicles to animals. And these vesicles will promote changes in the organ, and something that you can do is perform whole organ cultures of these tissues, collect liver-derived vesicles, and characterize the protein content. And something that we found, I mean, now we are pursuing more, is we found this protein here, like CORDC1, and that is more present in vesicles coming from pre-metastatic livers than healthy livers. And something that's interesting here is that this protein was already related before with metastatic dissemination and even liver metastasis. So this is something where we are studying right now, like whether we can use this protein in here that we identified as a way to detect liver pre-metastatic niche setup. Now, going back to prostate cancer, like something that, I mean, I think that is one step from what I just told you in the previous slide, is also trying to understand, like, I mean, for these guys, what they did, they saw that microRNA 407 that is present in fibroblasts that are, in this case, present in the microenvironment, tumor microenvironment of prostate cancer cell, both in the primary and the metastatic microenvironments. They found that these fibroblasts produce these microRNAs when they are activated, and these microRNAs, they could act in the prostate cancer cell, inducing proliferation and also inducing EMT features like staminase, for example. And they are suggesting that this could be linked with malignancy of those cells. And something that's interesting is, like, if you measure these microRNA in tissue sections, you can see higher expression of this microRNA in higher grade patients when compared to lower grade ones. And not only that, like, they could get this microRNA also by analyzing biofluids. So they could get access. The idea is that you could get access to the activation status of the fibroblasts associated to the prostate cancer by looking to this specific molecule in extracellular vesicles. Something that they didn't do, but I think that's something I would do, that is very tempting, is to test whether can we use this readout, the measurement of this molecule, to predict metastasis, or to measure modifications in bone microenvironment in prostate cancer patients. And of course, if you have this information, another big question is, like, what do you do with this information? But I think it's a very nice start point that can be done. People didn't do this yet, but I think that it would be very interesting. I'm getting to the end of the presentation. Like, something that we are also developing side by side with those projects is the technological aspect of these studies. Like, I mean, I told you that this is very easy, but I mean, easy if you have a lab with ultracentrifuges, our specialized equipment, the trained people, and time. Like, a lot of times, like, for you to get these results, like the measurements of those molecules that I told you, it may take up to a week or even more, depending on the methods that you are using. So something that we would need, that would be very helpful, would be a method that could perform a fast test of those readouts, and if possible, not using a lot of sample, because again, the samples are very precious, and the more tests that we can do with a single collection, of course, the merrier, the better, the more information we can get. So something that we are working in the group to try to get to this point is the use of vesicles flow cytometry. This is flow cytometers tailored to the analysis of very small vesicles, like as small as 100 nanometers. So for us is the perfect size to measure. Something that's also nice about this method, in addition to the other potentials, is they can provide you population information. Like this is not very common in the methods used to study extracellular vesicles. Usually people isolate those vesicles, they lyse them by billions, and they extract the molecules and then they study. But they don't know how many vesicles in this sample had the molecule of interest. This is something that can be done with this sort of method. Just to give you some preliminary results we have, something that's very nice is that it requires very, very little amount of samples. So something that is very useful is to use this method to study mouse models, like preclinical models, to try to go into the molecular biology of whichever question we are pursuing. And something that's nice, you can collect now blood from mice without having to seck them. In the previous method, you have probably to seck 4 or 5 mice to get one sample to measure. Here you can have all the animals alive during the whole experiment, collect samples them whenever you feel like, and analyze it longitudinally throughout. For example, here what we did was the injection of pancreatic cancer cells in the animals. We collected before the injection, 7, 14 days, and after the injection of the tumor. And you can measure these vesicles throughout the progression of the disease. This is pretty much showing us vesicles, the number of vesicles in the circulation of those animals. And here we used, for example, we measured like a tetraspanin that's usually present in exosomes, for example, just to show you that we can measure a molecule of interest also using this method. And something also very important, like especially for the clinics, like in addition to use very little sample, it takes only 3 hours for us to do this study. So like, for example, in Champalimaud, we have a pancreatic cancer fast track. The patients arrive usually early in the morning, and we try to have some conclusion of the exams by the end of the day. By having a method that can do the measurement in 3 hours, I think that now we can finally bring our studies to the clinical practice with this method. So here's just to show you, for example, another example, pancreatic cancer patients. This is a longitudinal study of the same patient, and the patient, in every point in here, the patient was evaluated by imaging to see whether the patient had progression or regression of liver metastatic disease. We could see here that here was the presentation when the patient arrived in the clinic. Here, the patient was having a successful chemotherapy. So they saw by imaging a reduction of the mass, and we saw a reduction of vesicles in the circulation of the patients. And then in the next point, the patient ended up losing response to the treatment, and the tumor started growing again. And we could see once more increase in the quantity of vesicles in the circulation of the patients. So this was just one example, but we can do it with even longer follow-ups, for example. And something that's nice is that every arrow that you see in here is a evaluation by imaging. So far, our method is working nicely to detect the points that patient had progression or response. And this is something nice in here is also, for example, this patient, and you find patients every once in a while like this patient, they don't show oscillations in another serric marker, CA99. So meaning that our marker could work as an alternative to follow up these patients for these patients that, usually don't show the oscillations that you usually expect from a classic marker. And this could be PSA, for example, in prostate cancer. I don't know. This is something that has to be studied. But I think even nicer than the previous slide, here we have also several points of follow-up for the patients. In addition to confirm the image, which is not very exciting, we had some evidences that, for example, the patient here was responding. And the patient had 1, 2, 3, 4 collection before the next imaging to measure the progression of disease or not. Something that was nice is that, I mean, 3 points before the imaging, we were already seeing increases in the levels of vesicles in the circulation. So this could be actually anticipating the result of the image. And this could be very important, of course, for the treatment of the patient. You probably drop a drug that's not working anymore and try to pick a next one that could potentially work better. I mean, instead of exposing the patient for a treatment that's not working, actually. And here, something that was nice, like when patient changed treatment in here, we also saw a decrease in these vesicles in the circulation, like when this patient had response and then he had progression once again. So so far, we are having very interesting results. We are expanding this. In addition to pancreatic cancer, we are also doing this with colorectal, with breast, and with prostate cancer patients. So we had some encouraging results that we are now pursuing. And to finish, like I told you about our interest in potentially targeting the pre-metastatic niches, right? We know, for example, in the lung microenvironment that one of the key molecules that play a role in the setup of these pre-metastatic niches in the lung, specifically in fibroblasts, is SRC. And we had very recently an opportunity to collaborate with Pfizer with the support of breast cancer now in a project that we are testing whether SRC inhibitors like bosutinib can be used to target SRC in lung fibroblasts. And our idea is whether we can use this to revert these metastatic microenvironments and to prevent metastasis in breast cancer. So this is something that we are pursuing also right now. Just like as next steps, as I told you before, something that we are exploring, but I think that other people should also pay a closer attention, is whether you can use tissue derived from tumor-associated cells, derived extracellular vesicles, like for example, lung, brain, bone, whether can we use them to early detect risk or incidence of lung, brain, and bone metastasis, as I was telling before. Something as we are doing in the lung, like whether once detected, whether we can target this microenvironment by understanding the molecular process that playing a role in the setup of this microenvironment, and of course to see whether we can prevent and revert metastasis by doing that. So this is the end. So this is just to show you the people who are doing all this work. We have a mix of clinicians, students, and postdocs in the group. So, and also something very important are the financial support and the collaboration with the clinicians that are not only providing the samples, but having a very important role in discussing the results that we have in our projects. So that's it. Thank you.
Thank you, Dr. Silva. There are a couple of questions here. If I may, I think we have time for another 5 minutes. So apart from the cancer-specific gene product, are there any way to tell the origin of organ from looking at the circulating or urinary exosomes, perhaps at the surface proteins of the vesicles?
This is a very important question. Like, I mean, there were some suggestions, like for example, GLP-1 for pancreatic cancer, but this is still very tough to my knowledge. Like we still don't have very good markers of origin. Something that we are doing actually, that not we, I mean as a field, right, but my group as well, is the closest that we can get to this answer right now is to, for example, studying DNA in those vesicles, because these vesicles also have double-stranded DNA. And something that's marked in cases of cancer is packaging of this DNA in vesicles. So if you find DNA positive vesicles in a biofluid like urine or plasma, this could be a very nice suggestion that these vesicles are coming from the tumor.
Okay. And maybe one more. So there's a question from Amin Ali, which says, is there a difference of EVs between different metastatic sites? For example, bone versus organ, or even primary tissue?
So if you're asking about, my understanding is that you're asking about the vesicles coming from the tumor-associated cells. That we don't know yet, because I mean the studies on that is pretty much, I mean, unless there are other people also doing, but we are one of the first to be doing that, to characterize like in what we're doing right now is actually comparing a healthy liver or versus a premetastatic liver. We are not even in the point that we can say, oh, this is coming from the liver, or this is coming from the bone, or this is coming from the lung. We're not there yet, but this is the idea.
Okay, yeah. One more. Okay, metastasis occurs also through migration across the perivascular bed. What is the role of exosomes in this way of tumor shedding rather than through EVs in blood circulation?
One of the things that I mentioned in the beginning, but I was very brief, is that something that they do in relation to the vasculature that is probably helps the metastatic process is the induction of vascular leakness. So we saw it very marked, especially in the cases of the lung. So these tumor-derived vesicles, the metastatic ones, they, with a single dose, we could see already a very strong effect in vascular permeability in those future sites of metastasis. So they were playing probably also a role by doing that. In addition to the recruitment of cells from other tissues, that this could be also a very important effect that they are doing linked to metastasis.
Okay. Thank you so much, Dr. Bruno.
Mm-hmm.
Okay, if I can hand over to our next chair, please. It's Parmvia.
Yeah, hi all. So up next we have a joint presentation by Annelies from Erasmus MC in Rotterdam and Ingrid from GenomeScan in Leiden. They'll be discussing their patient-derived xenograft and xenograft-derived organoid model of castration-resistant prostate cancer. So whenever you're ready, Annelies and Ingrid.
Good afternoon, everybody. I hope everybody can see my screen now and you can hear me.
Yes, we can.
Okay, thank you. Good afternoon. I'm Annelies and I will take off with our presentation, and then halfway through, Ingrid will take over to guide us through the bioinformatics part. So as Parmveer mentioned, I will show to you our novel panel of patient-derived preclinical models of castration-resistant prostate cancer. So local and systemic treatments for prostate cancer have evolved enormously in recent years, and a majority of patients can already be treated with curative intent, but nevertheless it remains a lethal disease because those patients that are presenting with advanced or metastatic or recurrent disease, they will eventually develop castration-resistant prostate cancer, which is still incurable at this time. So to broaden our knowledge on this lethal stage of prostate cancer and also to accelerate drug development for these patients, we need preclinical models that accurately represent the actual clinical disease and which are still very scarce at the moment. And we also want these models to be suitable for high throughput assessments. So prostate cancer cell lines have been instrumental for many developments in prostate cancer research, but unfortunately they are limited by some serious limitations as their overall very poor reflection of the actual clinical disease. While animal models, on the other hand, they way better retain the characteristics of the primary patient tumor, and they also more precisely predict the patient drug response. And that's also why our lab for many years now has taken the effort to generate human prostate cancer patient-derived xenografts. Unfortunately, these PDXs are limited to low throughput assessments, and to fill the gap between these 2D cell lines and animal models, we aim to establish prostate cancer organoids, and in that way circumventing some important biological and technical limitations of both models. So between April 2012 and December 2018, we have been collecting patient tumor samples from prostate cancer patients predominantly undergoing transurethral resections of the prostate, and we have engrafted these samples subcutaneously into intact nude ethemic mice to grow out as PDXs. So a total of 38 samples were collected, and from these 38, 9 showed evidence of tumor take. And from these 9, we were able to establish long-term PDX lines in 5 of them. So we next generated a workflow to create organoids from these PDXs, and for that, freshly resected tumor tissue was dissociated into single cells and small cell clumps. We were next culturing them in 3D to grow out as PDXOs, or PDX-derived organoids. We did this both for our PDXs that were already published before, and also now for our new PDXs. And these new PDXs we call the 2400 panel, because as you can see here, we have the 2400, 2412, 2416, and 2442 PDX. And then very recently, we have the 2459 PDX. This one was not yet processed to grow organoids from, because it's now at the mouse passage 7, and we will do this at the next passage. So I first want to focus on our PDXs. Importantly, all of these new 5 PDXs were derived from CRPC patients undergoing various pre-treatments. So we have androgen deprivation therapy, taxanes, and anti-androgenous abiraterone and enzalutamide. And I want to focus on these two because these two PDXs were actually derived from the same patient undergoing consecutive therapies at different disease stages. So our new PDX 2416 was derived when the patient experienced biochemical recurrence after androgen deprivation, so when he became castration resistant. And then the PDX 2442 was derived after subsequent treatments with docetaxel, enzalutamide, and cabazitaxel. So all of our new PDXs were pathologically assessed and showing adenocarcinoma with lumen formation in the 2400 and the novel 2459. And the other 3 in between, they showed evidence of large cell carcinoma or adenocarcinoma without any glandular differentiation. Importantly, as shown here by immunohistochemistry, all of our PDXs show express nuclear AR and also produce its downstream PSA. So we next wanted to test the response of our PDXs to castration. So what is shown here is in the red lines, these are mice that underwent a sham castration, and the yellow lines represent mice that were actually castrated. And you can see that upon castration, that tumors start regressing or stabilizing. For the PDXs from the same patients, we also investigated the influence of reintroducing androgens after castration. So again, in red, mice that were sham castrated, and then the yellow and the blue groups were castrated, and at day 30, they either received a testosterone pellet or an empty pellet. And as you can see, the blue group receiving a testosterone pellet showed immediate regrowth and progression of the tumor. So taking all of this data together, indicating that all of these new PDXs that were derived from CRPC patients are still driven by AR for their growth. And this is also reflected in the clinic, where CRPC patients need to continue their ADT despite castration resistance. So this is actually, these are some preliminary results. So these experiments are still ongoing, but we of course want to know the response of our PDXs to taxanes. And we first focused on the taxane response of the 24, 16 and 42 PDX. So the ones from the same patient. And as a reminder, here's the patient timeline in the right upper corner. So mice were randomized into 3 groups. In blue, the placebo group. In yellow, the group treated with docetaxel intravenously, and the red group with cabazitaxel. And as you can see, there was a survival benefit for both taxanes in the 24-16 with a temporal response, which was also seen in the patient. We're also testing the 24-42 at the moment, and as we expected, there is still some response to docetaxel, but with a shift and towards a shorter survival benefit compared to the 24-16. Surprisingly, this PDX seems to be extremely sensitive to cabazitaxel, and we are awaiting the final results of the experiments to further investigate these observations. So, as I mentioned in the introduction, from the PDXs, we made organoids, and we did this from our already published PDX panel, as you can see here. So we grew short-term organoids from most of the PDXs that we have in our lab. And then for the novel PDXs, we also grew organoids, and we maintained these in vitro as long as possible. Most of them were able to grow for more than 6 months and were kept viable during that time. All these organoids were characterized. And as you can see, they preserve nuclear AR, and they also produce PSA, and PSA was also detected in the medium that was overlaying the organoids. So, like I said in the introduction, we need models that accurately represent the disease and are amenable for large-scale assessments. So we first want to know, of course, whether our organoids that were derived from the PDXs, that they still accurately represent the PDX tumor that they were derived from. And for that, we performed RNA sequencing, both on the original PDX tumors as on the PDX-derived organoids, and these organoids were grown in 3 different media. And I will now give the word to Ingrid, who will guide you through the results on this. Ingrid, can you take over?
Yes, hello. So, as Annelies noted, the aim of my analysis was to evaluate the consistency between PDXs and their derived organoids based on transcriptomic data, and with the purpose of showing that the organoids are a valid representation of original tumors and as such can be used in further research. So the dataset consisted of 53 samples of bulk RNA-seq data. There were 7 PDXs, 3 derived from non-CRPC patients and 4 from CRPC patients, from which 2 independent samples were sequenced. This is seen in the left of the scheme. And from each PDX, 4 to 6 derived organoids were sequenced. So most of our organoids were 80% human based on the percentage of human reads after disintegration, which is in line with the expected amount of 20% supportive mouse stroma. We first analyzed the consistency between PDXs and their derived organoids based on variant profiles, after which we focused on gene expression profiles. The analysis so far used only RNA-seq data, but we will receive additional genomic data in order to validate it further. So here we see a variant profile comparison between the PDXs and their derived organoids shown with a heat map, where the PDXs are contained in the rows and the columns show both PDXs and organoids. The different colors in the legend indicate the patient they originated from. The similarity among samples here was calculated based on the intersect of variants that was scaled using quality scores from the variant calling. And the purpose was to show that the variant profiles of organoids are consistent with those of PDX tumors they were derived from, regardless of varying culture conditions. So the last 4 items on the scale indicate different growth media, which may cause selective pressure. So we see that the samples recapitulate the original tumor variant profiles, and this is also highlighted by the clustering of models from 2 independent samples of the same patient colored in blue and green. Next slide, please. This is another view of model consistency shown by a weighted PCA on the mentioned variant profiles. So the variance explained by the individual principal components was low, as you can see on the left, and this likely represents the variance of each patient, except for the case of the first principal component, which was found to correlate strongly with mutational burden. On the right hand is just an example view of the data separated according to the second and third principal components. Again, different colors represent different patients, different characters denote PDX status and organoid status. We got a very good patient-based separation of the PDXs and the organoids, which shows that they do represent the original tumors well, and that they are all consistent between each other in terms of variance. Next slide, please. So we next focused on the consistency of gene expression profiles between the PDXs and the organoids according to another weighted PCA. And similar to the previous PCA, the variance explained by the individual PCs was low and likely represents each patient. On the right hand is a view of the sample separation according to the first and third principal components. So again, we got a very good patient of origin-based grouping of the models, which shows that they are consistent with one another in terms of expression. And the models from the 3 non-CRPC patients grouped together as is visible in the top right corner. And it's actually interesting that models in black from PC2400, which was CRPC, cluster so close to those early stage PDXs. And this may be explained by some of the pathological features that Annelies observed. Next slide, please. Yes. So lastly, we decided to investigate the biology of the organoids which were derived from the same patient at 2 different stages of disease progression. Next image, please. The aim was to identify expression differences between the 2 stages and to see whether any of the differential expressed genes are known druggable targets that could explain the drug response of the patient that was observed. So in addition, we wanted to see whether some of these drug targets are amenable for drug testing to reproduce a drug response in vitro using the organoids. We conducted a differential expression analysis and identified a known druggable targets among the significant genes. And we then wanted to check which pathways linked to those druggable targets were perturbed. So I conducted a pathway enrichment analysis based on pre-selected sets of pathways, hallmark gene sets, and some other types of gene sets that contained some of those druggable targets that were most interesting. I did not show all of the data, but we identified numerous enriched gene sets, and this is an example shown here on a pre-selected set of KEC pathways where some interesting sets that were enriched include, for example, ABC transporters or steroid hormone biosynthesis. And this will guide our research and interpretation of the results further. And in addition to that, we are looking into transporters specifically. So next slide. In summary, 5 new PDXs were generated and matching PDX-derived organoids were established from them. These organoids seem to recapitulate well the original PDX tumors and resemble the complex tumor morphology more closely than traditional cell lines. And these organoids could provide substantial opportunities for future large-scale drug testing and screening in various stages of prostate cancer progression. Finally, we would like to thank our PIs and collaborators and acknowledge the consortium coordinator, Dr. Hing Leung. And thank you for your attention. Any questions are welcome.
Thanks, Annelies and Ingrid. So if there are any questions, just type them into the chat. I can start off with a quick question. So when you started your organoid cultures, what was the efficiency like and how long did they take to grow?
Actually, for PDX-derived organoids, they grow out quite quickly because you have a large tumor load. So within a few days to maximum 1 week, they can be passaged. I didn't mention it here, but we are also growing organoids from primary patient samples directly, so without first growing them as PDXs, and then it really depends on the patient sample. So some grow out very quickly, while others take way more time or don't grow out that easily. Okay,
and was the efficiency when you're going from PDX to organoid, was it about 100%? Was every PDX able to form an organoid culture?
Yes, all of them so far, except for one we've only tried once, but it's a PDX we're not using that very often. So that's one from the top of my head, that was the only one not growing out immediately. All the other ones were definitely feasible to grow short-term organoids from. And then the long-term ones we, for now, only tried from the novel panel, and they were able to grow for at least 3, and most of them at least 6 months.
Okay, that's great. So a question from the chat. The cell line highly sensitive to crumple taxol, what are the unique genes expressed compared to all other organoids? Also, is MDR expression different?
Yeah, so that's what we're looking into right now. So these results, so I think the one who asked the question was focusing on the 42. So this in vivo experiment is running at the moment, and with the differential expression that Ingrid is doing, we're looking into possible explanations why this PDX is more sensitive or very sensitive to cabazitaxel.
Yes, we did find a number of transporters that were dysregulated, and we are looking into them individually now.
All right, another question. You compared transcriptomics profiles between organoids and PDX. Did you also compare them with the original parental tumors?
The original tumors were sequenced in a different batch, and initially we had some comparisons, but we need to repeat this analysis with some batch correction.
Okay. Next question. In your patient-derived organoids, were you able to grow them up to 3 to 6 months? If so, did you manage to treat with taxane to see any difference with PDX organoid models?
Sorry, can you repeat from the word the primary patient-derived organoids or the PDX-derived organoids?
I think it means PDX-derived organoids.
So that's our next step indeed. So we first wanted to see the response of the PDXs to taxanes, and the next step will be to perform drug tests in vitro on the organoids derived from the same PDXs.
Okay, see if I missed any. For mice except PDX2442, sorry, for mice experiments, I'm assuming that's what that means. PDX2442 mentioned very sensitive to carbotaxol, but looked like a mixed response as 2 tumors regrew. Are these heterogeneous?
I will go back to that slide. That might be if I'm able to. Okay, so we're referring to this slide indeed. So the experiment is still ongoing. So for now, indeed, 2 tumors are regrowing, and I don't really want to draw any conclusions so far because it's still preliminary, and we have to see whether other ones are also going to regrow, because not all of them has reached the endpoint of 90 days yet. So it can be an issue of heterogeneity indeed, but I'm reluctant to draw any conclusions for now.
All right. And for how long can you keep patient-derived organoids in culture?
The patient-derived organoids were also, it depended on the patient sample. But for now, from the top of my head, we had about 15 attempts, and about half of them were able to grow for more than 6 months, but only a few were able to exponentially grow, and they can be used or will be used now for some drug experiments.
Okay. If there aren't any other questions, I guess we can move on to the next talk. So thanks, Annelies and Ingrid, and I'll hand it over to Anna, who's going to be chairing the next talk.
Hello. So I would like to welcome for today's symposium the early stage researcher Mina Sattari from University of Tampere, who is working on long-coding RNAs in prostate cancer. And today she's going to talk about the identification of long-coding RNAs with strong differential expression between primary tumors and metastasis of prostate cancer. So Mina, please, you can start your presentation.
Okay, thank you, Anna, for your introduction. And I would like to thank TransPot Consortium for giving me this opportunity to present my research work. I'm working on identification of a novel long-coding RNA with aberrant expression in prostate cancer metastasis. Okay, So prostate cancer is a leading cause of cancer-related deaths among men. While many prostate cancers are indolent, in around 20% of all the prostate cancers, and initially hormone-sensitive prostate cancer will progress to a lethal castration-resistant prostate cancer, or CRPC, which is one of the key problems in the clinical management of this disease. So there is an urgent need to have biomarkers that are able to distinguish metastasis-prone prostate cancers. Of course, it will enable better informed decision-making as to the course of treatment. So what are the long non-coding RNAs? Long RNAs are a novel class of RNA molecules. They do not encode proteins. They have a length greater than 200 nucleotides, and they play important roles in cancer. They can regulate cell proliferation, migration, apoptosis, and so on. And many studies have shown that link RNAs can be a really nice biomarkers for different types of cancers, such as prostate cancer. In this study, we are aiming to find link RNAs with strong differential expression between primary tumors and metastases as novel biomarkers. So these are the main objectives in this study so far. So we identified differential expressed link RNAs between primary tumors and metastases, like between primary prostate cancer tumors and metastatic prostate cancer. So we wanted to understand the variability of the expression of link RNAs in metastases, and also we wanted to know how different patients and different tissue types can affect that variability. For the next step, we did transcription factor analysis. We checked the binding site of really important transcription factors in the regulatory regions of our significant link RNAs. And next, we investigated the androgen responsiveness of our link RNAs, but this time using RNA-seq from cell lines. The data that we are using in this study is an RNA-seq data collected from 105 samples from 34 subjects. We have 16 normal tissue samples, 7 primary prostate cancer samples, and 82 metastatic prostate cancer samples. We also used publicly available data sets. For the annotation, we used high confidence set of lncpedia.org. We also have PCATs included from our previous work included in these data. And these are two main papers that these materials have been previously used in them. So first of all, we wanted to see how this data set look like, how samples are clustered to each other. For this purpose, we did principal component analysis for the 6 patients that they had primary prostate cancer samples. So the colors are indicating the patients and the shapes are for conditions. So the main thing that this PSA plot tells us is that patient-to-patient variability is really huge in this data set, is really big. Because as you can see, samples are clustered mainly based on the patients instead of the condition. So in this pipeline, I'm showing you what I've exactly done to get my differentially expressed links. First of all, I need to mention that working with link RNAs is not the same as working with protein coding genes. While everything is known for protein coding genes, their structures, their coordinates, their exon numbers, intron numbers, and everything. But here we are dealing with a novel class of RNAs that we don't know too much about them. And also, even though we are using a high confidence set of an annotation file, but still there is a lot of other things that may or may not be like correctly there. So these complexities can affect on how we look at these data, how we filter it, and how we analyze it. So for differential expression analysis, we use DSEQ2 bioconductor package, and we found 524 significant link RNAs. We have different criteria here for filtering this set. First of all, we remove the false positive link RNAs from the result, because link RNAs can have a tissue-specific expression. So by tissue-specific controls, we removed the links that their differential expression was due to tissue specificity, not because of the metastasis. And next, we did pairwise analysis. For the pairwise analysis, we checked the expression level of all the links in the 6 patients that they had primary samples. And the links that their expression showed a different direction in at least in more than 2 patients were filtered out. And next, we did some overlapping analysis. we wanted to remove the links that they have overlapping with the other protein coding genes and also pseudogenes, because we want to make sure that the reads in the count data belong to our link RNAs, not to the other stuff. But even though we did this overlapping analysis, I still checked all the results in IGV. I visualized them by IGV. For example, there were some link RNAs that even though they had overlapping with pseudogenes, but I could see clear patterns in the reads for my link. So it could tell me that those reads belong to my link RNAs, not to the pseudogenes. So I kept those. So after these filtering things, I now have 42 link RNAs, 36 upregulated and 6 downregulated. And I plot a heat map for these 42 differentially expressed link. Of course, this heat map is not perfect, and you can see a clear pattern here, but still, even though the effect of patients, effect of tissue types is quite strong in this data set, but still the downregulated links that here you can see them with a red box around them are close enough to each other, they are clustered somehow in the metastatic samples. And next, we did transcription factor analysis. For this purpose, we used previously published chip sequencing data sets, and we checked the binding site for AR, HOXP13, ERG, and FOXA1 in the regulatory region of our 42 significant link RNAs. And as you can see, about half of the links have binding sites for AR, HOXP13, and ERG, and there are 7 link RNAs that they have binding site for all these 4 transcription factors. And using Fisher test, I also calculated the p-value for these binding sites, and here you can see the p-values. Okay, and next we wanted to investigate the androgen responsiveness of our link RNA. So among the links that they had binding site for AR, we took them and we took a look at their expression in the cell line this time. We have RNA-seq from LNCAP and VCAP with AR knockdown and DHT stimulation. And the KLK3 or PSA has been used as a control here. And as you can see, for example, our link RNA, how nicely shows upregulation in the cells with 10 nanomolar DHT compared to the control in both cell lines. While in the siRNA-mediated treatment, AR knockdown treatment, you see a downregulation for our link RNA. So we can somehow claim that this link RNA is an AR-regulated long-non-coding RNA. Okay, so as a conclusion, we did a comprehensive gene expression analysis in order to get differentially expressed link RNAs between primary prostate cancer and metastasis. So as a result, we found 42 link RNAs. We have 36 upregulated and 6 downregulated. And among these 42 link RNAs, we have 7 previously identified links and they are published, and they are really essential for specific cancer types, such as prostate cancers. And also our transcription factor analysis revealed that about half of the significant links have at least 1 binding site for important transcription factors in prostate cancer, such as AR, ERG, and HOXP13 in their regulatory region. And also we found a couple of links that they show the higher expression level in LNCAP and VCAP cells with DHT stimulation, while they show the lower expression level in cells with AR knockdown. And as future plans, we are aiming to do a differential general transcription factor analysis. Like we want to do transcription factor enrichment analysis, this time using GTRD database, which is a huge database and it has many transcription factors, and we are aiming to see which transcription factors are going to be enriched in the regulatory region of our significant link RNAs compared to all the other link RNAs, so we can know more about our links. And as the next step, we are aiming to do ataxic analysis in order to investigate the accessibility of chromatin, and which helps us to identify promoter regions and potential enhancer and silencers, and it helps us to know how the regulatory mechanism for the gene expression for our link RNAs. And as the next step, we also would like to identify the prognostic values of our really most interesting link RNAs. And for this purpose, we are going to use RNA-seq from our prostatectomy cohort, and we can directly there ask questions whether any of these link RNAs somehow associate with prognostic value or not. And thank you for your attention. I would like also to thank my supervisors, all the members in our research group, and many thanks to TransPot Consortium.
Okay, thank you, Mina, for the nice presentation. Please, if you have any questions. Okay, there is one already. So first question, within your metastatic group, did you have both hormone sensitive and castration resistant patients within the cohort? If yes, was there any significant difference between the two groups?
Well, we had some hormone naive also samples in our group, but I haven't analyzed them yet. Like we are only using these metastasis prostate cancer samples from the patients, which which they have been, I think, previously treated.
Mm-hmm. Okay, so next question. Do you know if some of your link RNA are detected in circulating vesicles?
Circulating, I haven't checked them either, but that's a good question. I can also check them to see if any of them can be found in the vesicles or not.
Mm-hmm.
Yes.
Okay, so patient samples consist of other than epithelial cells. Do you expect your link RNAs to be exclusively cancer cell derived, or may they be linked to other cells or extracellular vesicles?
Well, if I understand your question well, do you asking that if we are sure that those cells are metastatic cells or not? Because we did some analysis to make sure that our samples are pure. So we are sure that those are from the cancer. Those are the cancer cells.
Mm-hmm. In the sample, there are also stromal cells. Dr. Vitsje explains. So.
Okay.
Mm-hmm. Okay. Until we have more questions from audience, I can also ask a few things to you, Mina. So are you planning to do any functional analysis? And if yes, based on what criteria you will choose your candidates for the functional analysis?
Well, that's also a good question. We still are not quite sure about our future plans, because our future plans would be like based on the results that we are going to get. But yes, doing the functional analysis is also one of the things that we have in our mind. We want to do that. For doing functional analysis, we are going to select the links based on their expression in the cell lines. So the links that they have a higher expression level in the cell lines are going to be selected for functional analysis. So we can do some overexpression analysis or silencing analysis to know more about our link RNAs.
Mm-hmm. Okay, thank you very much. So next question is: Are there long-coding RNAs that map in known prostate cancer protein coding genes?
Yes, I could find some link RNAs that they map with other protein coding genes that they are not prostate cancer specific genes.
Mm-hmm. Okay, and there is a comment as well. In the last issue of Cancer Research, an atlas of long-coding RNAs was published in many cancer and normal tissues. If not already, it would be helpful to check into that as well.
Oh.
So that was a comment. And another question from Dr. Vitsje: Our CRPC PDXs and PDX organoids all express AR. Do you think they will be useful validation tools?
Yes, of course they would be really useful, because we still want to somehow validate the AR regulation of our link RNAs. Of course, it would be quite nice.
Okay. And there is another question from Hing for your top long-coding RNA. What are the likely targeted genes, and do you plan to examine their expression in metastasis?
Which kind of metastasis? What? I couldn't understand well the question.
I guess he means in metastatic, your metastatic samples.
Any specific genes, check the expression of any specific gene on our metastatic samples.
Mm-hmm.
If there are some specific genes in prostate cancer, so that would be interesting also to check their expression in our metastatic samples. Of course, it can be also validating, like we can make sure that our samples can also express the genes that they are specific to prostate cancer samples. And please, if you have any suggestion, let me know. I can check their expression in our samples.
Mm-hmm. Okay, thank you. And another question, could you confirm that principal component analysis 3, first prostate cancer long-coding RNA is down in CRPC? Maybe you can turn in the slide where the principal component analysis is to explain that.
Okay, what was the question again?
Could you confirm that PCA3, I'm not sure what the...
Yeah, it's another...
It's a long-coding RNA, okay. Yeah. Is down in CRPC.
I haven't checked the expression level of PCA3 in CR, like in my samples. Like that would be that would be also interesting to see what's the expression level of PCA3 in metastasis samples compared to the primary. Yes, I will also check that one. Thank you for the question.
Mm-hmm. Okay, and can we check long-coding RNA expression using our GenomeScan RNA-seq data? Maybe Ingrid knows, so if you want also Ingrid to participate in this.
Yes, I think that was planned as one of the future steps for the paper. So where there should be some non-coding RNAs in our data, even though it was poly-A pulled down, there should be some. It's on my to-do list.
Yes, that would be interesting.
Yeah, so there's a significant portion of the non-coding RNAs that have a poly-A tail, so that is certainly possible.
Okay.
Mm-hmm.
That would be good.
Okay, so I have a last question for you. So regarding the ATAC-seq analysis that you explained in the end in the future plans, do you have ATAC-seq data from the same samples as RNA-seq data?
Well, no, we don't have ATAC-seq for the same sample, but we have ATAC-seq for our Tampere prostate cohort ready and available, and we can use that one.
Okay.
Yeah.
So are there any more questions? Okay, so we are 5 minutes ahead of time, I think, and now it's time for a 15 minutes break.
Thank you, Anna. Yeah, if we take our break now, so as Anna said, we're a little bit ahead of schedule. We'll take a 15-minute break. So if we can meet back at 1:50 or 2:50 European time, so 10 to the hour, and we'll continue with our next session. So see everyone in 15 minutes. Thank you. Okay, welcome back everyone. I'd now like to hand over to our next chair, Syed Umbreen.
Good afternoon, everyone. So the first speaker of our afternoon session is Teijo Pellinen. He did his PhD in biochemistry and molecular biology from the University of Turku in Finland, and at the moment is working as a senior researcher in the Department of Pathology, Tissue Imaging and Biomarker Research at the University of Helsinki in Finland. And today he's going to talk about the multiplexed imaging for tissue phenotyping and biomarker discovery. Over to you, Teijo.
Okay, thank you very much. And it's a very pleasure to be here with this prostate cancer audience. So in my presentation, I was thinking of focusing on prostate cancer tumor microenvironment and also do some comparison to other tumor types as well. So let's start. Okay, so recently we have developed a fluorescence-based multiplex IHC tissue staining and imaging method for both fresh frozen and FFP tissue specimen. It's also suitable for cell pellets and organoids. So here the kidney tissue works as a great example of multiplexed imaging example of different tissue compartments and cell types being just located next to each other in the nephronal system. And here we have stained nuclei with TAPI SOX9 for regenerating epithelial stem cells, alpha racemase for proximal tubules, which are also positive for CD10, and to some extent for vimentin as well, which mostly labels the glomeruli, however. Then E-cadherin for more distal tubules, and K67 for proliferating cells. So, recent upcoming of several antibody-based multiplex imaging technologies have taken place. This is a little bit of a summary of the different methods, and the ones on the right-hand side have a significantly higher level of multiplexing as compared to the technologies on the left. The ones on the right are suitable for staining handful of samples due to slow processing and high costs. But however, they are great, for example, for deep molecular profiling of tissue samples. However, here the ones that we have developed and others is not the most highly multiplexing, as we cover only up to 7 targets at a time from the same tissue sample. However, it allows spatial analysis of hundreds or thousands of patient samples in a few days with low costs. So this is more suitable for large-scale prognostic studies. And I would say that these are not really competing technologies, but I would rather like to see them complementing each other, that, for example, early discovery is done with high multiplexing and then prognostic studies later on with the less multiplexing but more high throughput ones. So we do the imaging of samples using one of these high resolution ultra fast scanners. The imaging is based on fast bandpass filter system, and the special to these are that we have equipped all these machines also with near infrared fluorescence channel, enabling us to detect 5 channels without crosstalk from one channel to another. Also important to notice that in the higher wavelengths, there is much less autofluorescence, and in fact, in the NIR, there is really none autofluorescence. So we have a 5-channel imager. How do we then capture 7 markers or 7 channels? So in 7-plex staining, we first stain and image 5 channels with 4 antibodies and DAPI for nuclei. And the 2 lowest wavelengths are TSA amplified. Due to high autofluorescence in this region, they need to be amplified. Don't even try to do 488 and 555 signal detection without amplification in FFPE tissue. Then after this step, we heat denaturate the sample and perform a second round of staining and imaging, now with the two uppermost channels for the antibodies, as the TSA ones cannot be reused. We then bleach the uppermost fluorescent signal and do another cycle of staining. And lastly, the images from different cycles can be registered, meaning overlaying on top of each other using the common nuclear DAPI signal. So this is the technology in short. So we have optimized now more than 50 immune cell markers to cover different types of immune cells with examples of the different antibody panels shown here. And here is an example of ovarian cancer stained with the hot topic checkpoint inhibitor antibodies PD-1, PD-L1, and then macrophages and epithelium. Why I bring this is to show the benefits of multiplexing, that typically the PD-L1 is detected or analyzed within the cancer epithelial cells, but often there are immune cells in the epithelial or within the epithelial compartment, causing trouble in single color staining evaluation. But with this technology, you can easily discern whether it's macrophage or other cell type within the epithelium. This is just an example of strong PD-L1 expression in lung adenocarcinoma tumor compartment. Okay, so the digital image analysis we do is based on cellular segmentation based on nuclear DAPI, allowing us to classify cells according to their marker expression levels in a cytometry-like fashion. Another option, which is much less demanding for the computing, is to do big pixel-based analysis. In the pixel-based analysis, we classify the pixels instead of cells and measure the relative areas occupied by different pixels. So often in imaging world, people are arguing whether one should use cell-based or pixel-based analysis. And I've been doing quite a lot of comparison between these methods. And I have to say that the pixel-based is often even better than cell-based, although it's sometimes difficult then to talk about cells as units, but as pixels, which is not understandable for many in the audience. Okay, so recently, by using both pixel-based and cell-based image analysis, we found that cancer-associated fibroblasts, classified as positive for vimentin, but negative for pancytokeratins and E-cadherin, strongly predicted cancer-specific survival of prostate cancer patients in 2 different prostate cancer cohorts. So now, motivated by this, we initiated a project of pan-cancer stroma profiling, where the idea is to do spatial imaging of selected fibroblast and immune cell markers across 15 to 20 different tumor types. So as a background, the cancer-associated fibroblasts do affect tumor progression by modulating immune cells, both by supporting and suppressing their functions. The general idea, however, is that CAFs dampen the anti-tumor immunity. However, there's a lot of heterogeneity in CAF markers, and thus it would be very important to better characterize different subsets of CAFs in different cancers. The overall hypothesis of this project is that CAF subsets could be developed as drug targets and biomarkers of clinical utility, perhaps in combination with immuno-oncology therapy. So we are currently profiling selected panels of CAF and immune cell markers in more than 15 different tumor types, each having hundreds of tissue samples with clinical follow-up data. And now I will give you examples of the preliminary data we have obtained from profiling of prostate cancer and with comparison to lung cancer. And I will also discuss more about the prostate tumor microenvironment with respect to magnetic resonance imaging based diagnosis and risk stratification. So in this staining, we have stained 6 markers plus nuclei from the same sample. We have just started this first CAF panel profiling, which has antibodies for different fibroblast markers, so patient-derived growth factor receptor alpha and beta, fibroblast activation protein, alpha smooth muscle actin, and then epithelial stain, which is combination of 3 different clones of antibodies. And then in the prostate case, we have also included basal epithelium for detecting the benign glands. So after staining and high-resolution scanning, we performed pixel classification using elastic machine learning to recognize and detect epithelium and stroma, and also the lumen areas. And then we classified each pixel in the stromal segment as positive or negative for the 4 CAF markers, giving 16 different pixel classes whose areal fractions could be measured in the total stromal segment. So we have now stained and analyzed a radical prostatectomy cohort of 304 patient tissue with replicates, and including the records of cancer-specific follow-up clinical data. And in addition, we have the genomic profiling and immune cell marker profiling ongoing for this cohort. So what came out using Cox regression survival analysis, we found that single CAF marker phenotype status did not predict the progression or survival of prostate cancer patients. Here, the marker positivities in cells is done with a median cutoff, but the same results were seen using continuous digital values. However, analysis of the 16 multi-marker phenotypes yielded a significant survival effect for one phenotype, namely PDGFR beta positive, alpha negative, FAP positive, and SMA positive fibroblast phenotype. And here on the left, an example of the prognostic phenotype with the low phenotype, and then the high CAF phenotype, and the associated Kaplan-Meier survival curve with median cutoff dichotomization. Okay, then we went to the non-small cell lung cancer cohort to do a comparison of prostate cancer with lung cancer. And this cohort has relapse-free survival data, but also, importantly, genomic and immune cell profiling done to make associations with the fibroblast markers. Here an example of the first round staining, and in the second round staining, we actually replaced the CK5 with an endothelial marker, CD34. Similar to prostate cancer, a single marker positivity did not stratify patients to poor and good prognosis groups. However, again, the very same multi-marker phenotype as in the prostate cancer cohort predicted disease progression. And this was also validated using continuous values. Now here, an example image of the low CAF phenotype and high CAF phenotype in lung cancer. And interestingly enough, we found that this particular phenotype was prognostic only in the patient group with wild type TP53, but not with TP53 mutant status. This serves as a very nice example of how a combination of a genotype and a phenotype may give additional value to the prognostics. We also found that this same CAF phenotype correlated with immunosuppressing tissue phenotype with higher numbers of FOXP3 positive Treg cells, CD163 positive M2-like macrophages, and PD-L1 positive tumor cells. It's remarkable that this positive correlation we found with immune suppressant phenotype is in line with earlier findings from the group of Mehta Grigoriou, who showed that PDGFR-beta, FAP, and SMA positive CAFs in breast cancer confer immune suppression by attracting FOXP3 positive Tregs. Now the results together suggest that targeting this type of CAFs alone or in combination with immunotherapy should be investigated, at least in breast and lung cancer, but we are now investigating this association also in prostate cancer. So now I jump to the magnetic resonance imaging and the stromal cell biology affecting the diagnosis of MRI. So magnetic resonance imaging is a popular method to detect and help in diagnosis of prostate cancer. It has improved the preoperative prediction of clinically significant prostate cancer. However, many studies point out that a substantial fraction of clinically significant cancers are missed at MRI. Now, the molecular characteristics between MRI negative and positive tumors are not known. However, Rosengratz and others have demonstrated that MRI visible tumors always contained desmoplastic stroma. The desmoplastic stroma was defined as stroma with increased size and number of fibroblasts with rich capillary network, often with mixed inflammatory cell infiltrate. In this study, we compared prostatectomy tissue with patient-matched samples from MRI missed and MRI positive tumor regions. And as a comparison, we also included matched benign tissue regions for the analysis. In this study, we stained the FAP and SMA, but also CD8 for T killer cells and CD163 for M2-like macrophages. What we found was that the number of SMA positive cells decreased from benign to MRI missed, and again from MRI missed cancer lesions to MRI positive lesions. This was totally opposite for FAP positive or CD163 positive and CD8 positive cells. Especially the FAP positive cells were much more abundant in the MRI positive cancer lesions as compared with either matched MRI negative lesions or benign tissue areas. In a Cox regression univariate analysis for the preoperative variables, Capra risk grouping predicted BCR, so biochemical recurrence. None of the MRI variables predicted BCR. Of the post-operative variables, Gleason grade grouping, capsular invasion, seminal vesicular invasion, and pathological staging predicted BCR when corrected for multiple testing using Bonferroni correction. Then importantly, when looking at the different stromal marker-defined cell populations, higher FAP positivity predicted earlier recurrence, whereas this was opposite to SMA, which higher expression predicted longer recurrence-free survival. It's also of note that these cell types were prognostic only when measured within the MRI positive cancer lesions, but not in the MRI negative lesions or benign tissue areas. This example image A shows high FAP staining in between tumor glands in blue color. It was clear that FAP distribution was not normally distributed in patients, as about 90% of the patients were almost negative for FAP positivity or for FAP. Kaplan-Meier analysis demonstrated that patient dichotomization, either with highest FAP decile or median cutoff, showed clear prognostic effect. The example in E shows high SMA positivity and a more normal distribution across the patients for the SMA. A median cutoff dichotomization yielded a significant prognostic effect for SMA, but to the opposite direction than FAP. When adjusting for the most important clinical variables in a multivariable model, high FAP positivity was independent prognostic factor. Also, when taking into account the SMA positivity in this model. And finally, it was very interesting and in line with our previous results in lung cancer, the FAP positive cell fraction correlated with CD8 and CD160 positivity in this MRI cohort. And interestingly, we also measured the physically interacting FAP cells. About 25% of all the FAP positive cells interacted with CD8 positive cells. So this interplay between cancer-associated fibroblasts and immune cells is very interesting and could be the target of therapy. As a conclusion, I would say that multiplexed IHC-based marker profiling is a great tool for high-throughput prognostics, for the discovery and validation of cell subtype associations, and for creating novel hypotheses of functional interplay in the tumor microenvironment. In the future, it will be highly interesting to find out if there are more of these common or more unique stromal phenotypes associating with disease progression across different cancers. And with more sophisticated image analysis tools, we are currently also analyzing cell subtype spatial distributions and how they may associate with disease progression. I would like to thank my mentor, Professor Olli Kalliniemi, and the associated group members with whom I work for the multiplexed imaging of tissue. And for the prostate cancer studies, I work in close collaboration with a pathologist, Tuomas Mirtti, and a urologist, Antti Rannikko. Thank you very much for your time.
Thank you, Teijo, for your presentation. That was great. Any questions, you can please type in the chat. So I'll start with a question. You mentioned about denaturing your slides with heat after the first round of staining. Does denaturing with heat affect the integrity of the tissues that you're working with?
Well, if you do many cycles of heating, then definitely you get unfolding and some artifacts to the tissue, which is not nice. So that's why we don't continue too many cycles, but we typically use 1 or 2 staining cycles only. But the heating step is typically done also in normal IHC, so heating is in fact necessary to penetrate the tissue for antibody staining. So a little bit of heating you need, but too much is really unfolding tissue as well.
Right. So we have some questions in the chat. The first question is, with prostate cancer known to have intratumoral heterogeneity and always multifocal, did you just had a look at index lesions or multiple lesions?
Yeah, this is a very good question, and I think the heterogeneity can be so that you have many index lesions in prostate cancer. Some of the cohorts we have used here, the population-based so-called Helsinki cohort, is based on taking 3 different cores from the most representative Gleason area, the second Gleason, and the third representative Gleason area. And I don't think at this point pathologists and clinicians really thought about the multifocal disease so much. So I don't think that we can answer to these questions using these older generation TMAs. But in this magnetic resonance imaging study, we do have all the locations of the lesions marked and where we have analyzed also using multiplex imaging. But for this study, we didn't do detailed analysis on the spatial locations, but will be the topic of the future studies, yes.
Thank you. So the next question is from Utski. Her question is, do you use pixel or cell-based analysis for these clinical evaluations? And when you use pixel-based analysis, how do you correct for membranous versus nuclear staining?
Yes. So the first part of the question, can you repeat it?
Do you use pixel or cell-based analysis for these clinical evaluations?
Well, most of the studies we have done is by using pixel-based analysis.
How do you correct for membranes? Yeah.
Yes, and why we do is that we haven't thus far. So it's really thus far. We haven't found good cell nuclear and cell membrane segmentation tools, but just recently with deep learning, we are getting into that. So we are changing our pixel-based methods towards cell-based. Also, the cell-based method requires much higher computing levels and a lot of time also. So we are moving towards cell-based, but still I would say that often the pixel-based and cell-based, they correlate very well. And then what comes to the nuclear or membrane staining, we cannot really define whether it's nuclear or membrane or cytoplasmic staining when doing pixel-based, only to the limit that we have each pixel also, we can classify it positive or negative for the nuclear marker DAPI. So we can see whether it is co-localizing with the DAPI nuclear signal. So that much we can say whether it is in the nuclei or not using the pixel-based analysis.
Thank you. The next question is from Hing. Can you comment on the underlying molecular reason for FAP-positive CAF with CD8 cell? I'll repeat. Can you comment on the underlying molecular reason for FAP-positive CAFs with CD8 cells? That's
a very good question. I've been thinking about it a lot, and I really do not have the correct answer to that. I think we would need to look at the CD8 cells in more detail, what other markers they do have, and especially other lymphocytes as well. It could be that these are somehow immunosuppressing cells as well. They could have some exhaustion markers present. And typically the immunosuppression and exhaustion markers are derived from the microenvironment, such as different cytokines and growth factors. And I have had this idea that this could be the effect of TGF-beta that is known to induce the expression of FAP. It's known to induce the expression of T regulatory cell markers. It's known to induce the expression of M2 macrophages. And in the future, we are working on looking at TGF-beta signaling in relation to this immune cell CAF correlation and co-localization. So that's my best guess at the moment.
Okay, thank you. Hing has one more question about the lung cohort that you talked about. It seems that P53 mutant cases have a slightly better outcome. Can you comment, please?
I don't totally agree, because I've been investigating these curves. I don't have it here now open. I could open it, but the TP53 mutant cases compared to TP53 wild type cases, the TP53 alone as a mutation is associated with worse prognosis. And in fact, in this cohort, this has been already published. And but what was happening was that with the TP53 wild type, you could see the discrimination or the separation of the two patient groups with the CAF phenotype, and this might give you kind of a false view of the global situation. But it's another question then, what is happening with the TP53 and this CAF phenotype? And I would really love to look at this also in prostate cancer, although it is known that in localized prostate cancer it might be present only in 5 to 10% of the patients. So I don't know whether we can get easily any associations, but if anybody in this audience has a good cohort with TP53 mutation status, please contact me and we can collaborate on this.
Okay, thank you. I think we still have 8 minutes, so we can pick up a couple of more questions. We have a question from Matthias. So his question is, how long does such a 7-plex staining protocol take? And do you have a large amount of loss of tissue?
How long it takes, let's take a typical prostate cancer TMA, like we have the Helsinki and Turku TMAs. They have 25 to 35 slides each. We typically stain 30 to 40 slides at a time. We can also put it in the machine. So machine does the first round and then we take it out. We do the heating manually and then we do the second round in the machine. So in 1 week time, we have done the 3 cycles for 30 slides or 40 slides, whatever we take for one staining. So 40 slides in 1 week is a reasonable time. And then imaging takes a couple of days for these.
We have one more question from Vitsky. She might have missed it. Her question is, but is there no link with vascularity? Do you have functional MRI to link with these stainings?
With vascularity?
Yeah.
I don't know about any link with vascularity. It could be that I have, if I don't remember totally wrong, I have seen something about vascularity and MRI. But typically, if you have more water molecules, if you have more luminal space, you have that affecting the ADC value in the MRI. So anything to do with water molecules, and this also means the cell and tissue density has an effect on MRI results. So for example, MRI typically associates with lesion score and the grading of the tumor because the cellularity, glandularity is different. Vessels, I would think that there is some association with the ADC values of MRI, but I can't recall.
So so Hing has a question again, following on his earlier question. Now his question is, did you specifically look at data from DCE, dynamic contrast enhancement with correlative analysis?
This is related to the MRI, and I'm not totally aware of what was the main readout in the MRI. I know that in Helsinki they used 3 different readouts and a combination of these. So I cannot really answer whether it was specific to certain readout or whether it was a combination of readouts. This is more of the clinicians or radiologists' specificity. So I cannot answer to this question.
Okay. And one last question is from Amin Ali. His question is, what's the longest archival tissue you have used which did not compromise the quality of your multiplexing imaging?
Well, we have paraffin blocks from 70s and 80s, and they seem to be fine. It seems that you just have to trim out some micrometers from the surface of the paraffin block. So paraffin blocks are great for storing tissue material. It's great also for cell and organoid material storage, because you can come back to your models, to your samples later on and ask different questions than initially what you were planning. So the biggest problem is that if you cut the paraffin sections and store them long time, we can see effect with some antibodies after a couple of weeks. PDL1 is one of those that you really have to do staining within 1 month time maximum. Some markers, like the stromal markers, don't really care. They can be on the slides, the sections, for half a year, and the stains look very similar. But you have to always be aware that that oxygenation or some deterioration happens for the samples, and be ready to be aware of that. Yes.
Thank you, Teijo. I think that was a wonderful presentation, and we took most of the questions from the chat. So I'll hand it over to the next chair. Thank you so much.
Thank you.
Thank you so much.
Hello, everyone. Can everybody hear me?
Yes, we can.
Okay. So my name is Rafael. I'm the ESR from University of Glasgow. First of all, good afternoon. And I'm going to introduce Mario Cangiano. He's another ESR, and he's based on the Netherlands. He's currently working at GenomeScan under the supervision of Ines and Bart. Mario is working to integrate prostate cancer datasets from several laboratories within the TransPot network, including our own. And he has a special interest in regulons and units of regulation that can potentially play a key role in prostate cancer. And today, he's going to talk to us about, as you can see, transcriptional co-regulators activity status as a prognostic signature for recurrent predisposition in a prostate cancer patient. So after that mouthful, I'll let you with Mario, and he can start spelling all his science to us.
Mario, we can't hear you. I think you're muted.
Hello?
Hi, we can hear you now.
Yeah, sorry, thanks. So, okay, I just say that, well, good afternoon to everybody, and thanks, Rafael, for the introduction. As you said, I'm the ESR number 9, working at GenomeScan as a computational biologist. Within my project, I'm focusing on the analysis of data coming from both preclinical models and patient cohorts in order to identify a prognostic signature that is able to, let's say, overcome the limitations of the standard of care and the standard also of the prognostic markers that are currently used in clinical practice. This because prostate cancer is currently treated with androgen deprivation therapy as a first line of treatment. But after that, virtually all patients progress into a status that is called castration resistance, namely when you can observe a biochemical or radiographic progression of the cancer, of the disease, despite castrate level of testosterone. So currently, prostate-specific antigen, also called PSA, is used as a marker to decide what treatment the patient should receive. But unfortunately, this comes also with a drawback, because there are many false positives associated with this marker. And then there is still a need, an urgent need of a more accurate prognostic workflow. I would like to start by introducing the datasets I've been working with and that I've currently been provided with from member of the consortium or partners of the consortium. So first of all, I'm working mainly on RNA-seq data. And in particular, I have one preclinical model dataset coming from the University of Glasgow, where I have 18 tumors that were injected into, that were derived, sorry, from autografts, from mice autografts, and 4 independent datasets of patients, cohorts, whose that were used as a validation or test for testing and validation of the results obtained from the preclinical models. So I want also to underline that these samples were all fresh frozen samples, and some of the patient cohorts had even multiple multifoci samples from the same patient. But just to be clear, in this analysis, all the multifocal samples were pooled into one in order to simplify the analysis. So here I'm describing the workflow of the pipeline. So first of all, the preclinical models, given the fact that the tumors were grown into nude and immune nude and either castrate or not castrate mice, they preserve, let's say, the biology of the prostate cancer without any interference from let's say also the minimum interference from the human stroma or the immune infiltration. The preclinical model's RNA-seq was used to generate a gene regulatory network, namely a graph structure that expresses and shows all the relationships that exist between transcription factors and their targets, namely the genes that are positively or negatively regulated. I want to also pinpoint that the rationale behind this analysis is the hypothesis that prostate cancer is a disease driven by transcriptional reprogramming. And that's why I wanted to focus on the activity of transcription factors, and especially how they are connected to each other. That's why I wanted to have an overview of all these interactions with a gene regulatory network. This was actually used as a basis for the enrichment analysis, which is called the gene graph enrichment analysis in this case, by using the differentially expressed genes calculated at a single sample level from the patient cohorts. So every patient cohort was used as a standalone dataset to calculate the differentially expressed genes that were then in turn used to enrich the gene regulator network to understand which transcription factors were active or not. Starting from this transcription factors profile, we wanted to check if there is any specific or couple or signature of transcription factors associated with the survival, and in our case with the biochemical recurrence, so with progression-free time. And we even had the chance to validate at Glasgow to have an in vitro validation of one of the candidates that came out from this analysis. So I wanted also to give some details about the statistics that has been used, because in this case, given that I needed a profile for each patient sample, I needed to calculate the differentially expressed genes at the per sample level, which is not the standard practice in bioinformatics. And so I did it by standardization of within each patient cohort of each tumor sample with all the panel of normals from the same dataset. In this way, you can get rid of any difference in experimental design and also in batch effects that you may have while comparing different datasets coming from different labs. So you can obtain a profile that is very specific for that is only revealing the biological differences. So using these differential expressed genes, as I said before, I obtained a profile of activity for each of the regulants that I've identified from the gene regulatory network. And we're talking about 1,300 regulants that were identified. And so for each sample, namely each patient, I will know which of the transcription factors are active or not active. And exactly this profile that you can see here in this matrix as columns were used for the survival analysis. So they were regressed against the biochemical recurrence data. And these are the main results in the 4 patient datasets that I've used, for which, of course, I had survival data. And what these curves are, Kaplan-Meier curves are showing is that when the candidate gene that we are still studying, transcription factor that we're still studying is active, then you have a worse prognosis. Sorry, I made a mistake with the legend. It's the opposite. So you have a worse prognosis. So you have the opposite when that specific regulon is inactive. And this actually is a robust result because it applies for all the 4 datasets. And we can also see that the hazard ratio is similar, is comparable. So that also is a kind of evidence of, let's say, support of this evidence. And of course, the trend of the analysis is the same in both the dataset from Tampere, in TCGA, in the patient cohorts of the Erasmus Medical Center, and in the ICGC cohort. So given that these results were promising, we decided to check the effect of the removal of such a transcription factor in one of the prostate cancer cell lines that were available at the Beatson Institute in Glasgow. And in particular, we were focusing on the CWR22, which are hormone-naive prostate cancer cell lines, and the correspondent castration-resistant cell lines, which are called 22RV1. Because again, the goal of this analysis was to show that this transcription factor is always somehow important in the progression and the evolution of the disease. And then we wanted to check if indeed we would see an effect of its knockdown with a short interference, a small interference RNA in both cases. And so within this experimental design, we checked if in the hormone-naive cell lines, both cultivated in standard serum, so fetal bovine serum, which contains testosterone, but also in CSS, which is a serum that instead is steroid depleted. We checked for the growth trend of these cell lines within 5 days after removal or knockdown, in this case, of the gene of interest. And actually the results were also nice because they show that in general you always see a difference with the controlled small interference RNA and the target one in both hormone-naive and castration-resistant environment. Then we also had a look at what's happening at the protein level. And we wanted to check if this protein was also somehow cancer specific. So thanks to the help of the colleagues at the University of Glasgow at Beatson, then we also had a panel of prostate cancer cell lines, some of which are hormone naive, others are castration resistant, plus an epithelial cell line from prostate. And we just demonstrated also with this Western blot, we had another evidence that this protein seems to be cancer specific because it's lowly expressed in the normal cell line. And that's why this motivated us in performing additional analysis that we are still performing. So with this presentation, I wanted to show that apparently a graph-based enrichment analysis, so by using the gene-gene interaction informations when it comes to transcription factors and their targets, provides a consistent and apparently an accurate prognostic marker for biochemical recurrence, at least at the RNA level. And that's why we need to do also further investigation on what's happening also at the protein level, and if we can validate these also in additional cohorts. But also the important thing, given that we know now the interaction of the genes, we may even decide to look into greater details of these transcription factor targets relationships to identify novel mechanism of PCA progression, or prostate cancer progression. And well, of course, these results, if they keep being that promising, could also be implemented one day in a personalized medicine treatment, in the sense that a specific group of patients who are showing the molecular marker active are probably predisposed to biochemical recurrence if they receive the standard of care, or namely the androgen deprivation therapy, and probably they could instead address to a different pathway, a different path of treatment. And yes, this was my presentation, but I also want to take the chance to thank all the members of the consortium, and especially the ones who helped me in both analyzing the data and in validating also my results at the bench level. Thank you.
Okay, thank you very much, Mario, for your great presentation. So from now on, if anybody has any questions, please use the chat and I will read them out for Mario to answer. So I will start with a question. Is so when you define regulons, sorry, my question is exactly if you define the regulons before doing the analysis, or do the regulons emerge naturally from the associations the data analysis finds?
Yes, okay. So the regulons derive from a prior knowledge, which is just the transcription factors that you want to investigate. And I decided to use the list from the molecular signature database of human transcription factors. But then the regulon themselves are derived from the RNA-seq data. So they are calculated based on the correlations of the expression of the transcription factor and the expression of the targets. So in this case, Glasgow data, so the preclinical model data was used for the inference of the regulons.
Okay, great. That was a great answer. I have another question from the chat. What's the correlation of your candidate gene with non-prognostic marker like Gleason score or diagnostic PSA?
Yeah, so, sorry.
No, go ahead.
Yeah, thanks for the question. Yeah, actually, I did not show this, but when I used the Cox regression to check for the association with biochemical recurrence, I also checked if let's say the significance is preserved, even if you consider Gleason score or other kind of markers like PSA and the tumor stage. But in my case, at least, it's in these 4 data sets, there was always significance even when considering Gleason score.
Okay, there's an addendum to that question. If have you also looked at a metastatic cohort?
No, actually not. For this analysis, I focused only on localized tumors. Okay. Who received radical prostatectomies.
Okay, another question from Hing. Are there patterns of regulation of transcription factor associated in a way that you can consider subtypes of tumors?
Yeah, I didn't check that actually, but that's a good suggestion. Maybe this will be a follow-up of the study.
Okay. So there is no more question. I'll check the time. I think we still have time. So I have another question. You use the Glasgow data to do the initial analysis, find out this candidate gene, and then redo the analysis on the rest. What happens if you do it the other way around? If you use every other data sets to generate a signature, do you find the same target, or do you find other prospective candidates as well?
Mm-hmm. Yeah, I didn't do that because I wanted to keep the dataset as clean as possible. Let's say the learning dataset. And given that preclinical models are in this specific case, are let's say free of any immune or human stroma interaction influence in the expression of the genes, then I prefer to use the Glasgow dataset. But this, okay, of course I could also try the other way around. I didn't do, but I expect, let's say, the regulants to be at least slightly different, because of course you may have a lot of, let's say, cleaning steps to do, pre-processing steps to perform to the data before you can get it clean.
Mm-hmm. Okay, what would be your key experiment to demonstrate how good your prospective gene is? What is, if you have the resources and the time?
Yeah, that's what's happening at the moment. So I would like to check if the protein expression, actually the immunohistochemistry staining of the protein in a TMA is also associated with biochemical recurrence. So in an independent cohort. That will be also a good validation also at the protein level.
Okay. So if there are no more questions on the chat, I'm afraid I'm also out of questions. You answered very well to my questions. So there's one from Whiskey. Did you check if this candidate gene is prostate cancer specific, or may be a resistant marker for other cancer types as well?
Yeah, apparently, well, it seems to be, the protein at least seems to be expressed in prostate cancer, but I don't know exactly what's happening in other cancers. But I found some, at least not in all of them, I found something also in breast cancer. So it could be, maybe it could be that it's also not very prostate cancer specific, but I still have to check that. Maybe I will look at the datasets online from TCGA.
Okay. So, any more questions from the chat? Last minute. Okay. So, if that's okay, I think we're a bit ahead of schedule. But Katie, if you want to, I can move on to the next talk and hand over to the next chair.
Yep, that's great. Thank you.
Thank you very much.
Thank you.
Okay. Hi, everyone. So our last ESR of the day is Marouane. Marouane is an early stage researcher working in Numaris Health in Germany. And today he will be talking about his research in the identification of urinary biomarkers in relation to prostate cancer progression and castration resistance in prostate cancer. So Marouane, begin whenever you're ready.
Okay, thank you, Gal, for the introduction. So, hi everyone. I'm working on the identification of urinary biomarkers in relation to PCA progression and CRPC. First, just go in presentation mode, and I will give you a quick background about the subject, although this was covered by most of the presentations today. So, as you know, the first line of treatment in patients diagnosed with advanced prostate cancer is androgen deprivation therapy, which reduces the levels of androgen hormones in order to slow the growth of prostate cancer cells. But within a median of 18 to 24 months, within a median of 18 to 24 months, most of patients relapse and develop what's called castration-resistant prostate cancer, which is basically the disease progression despite the treatment. This condition has limited therapeutic options, and also the molecular mechanisms behind it is still poorly understood. So the aim of my projects are to identify the metabolomics alterations between patients who responded to the treatment at this point and those who developed CRPC, and also to test the transferability of these markers to identify aggressive prostate cancer. At the end, we would like to integrate these metabolomics data with transcriptomics and proteomics data in order to reveal pathways and maybe other potential biomarkers involved in this castration resistance mechanism. So to this end, we obtained a urine samples from 2 groups of patients that then we split the sample set into 2 cohorts, one for the training and the other one for testing of the models. The data sets contain 2 groups of patients. The cases are defined as patients who have prostate cancer treated with the GnRH analogs, but they show clinical signs of tumor progression. So their testosterone level is below the castration level. They have a confirmed PSA increase by a factor of 100, and they also have a newly diagnosed bone metastasis. As for the controls, they are patients who have prostate cancer treated too, but they are in stable remission. And this is characterized by testosterone level below the castration level and also a low value of PSA, and they don't show any clinical signs of tumor progression. So I will walk you through the methodology used in the course of the project. First, we measure the urine samples of these patients using NMR spectroscopy. Then we obtain the raw spectra of each sample or patients, and then this spectra is processed. The processing step is very helpful in order to check for the quality of the spectra and also the comparability of the spectra between each other, like normalizing the spectra to the creatinine. Once this is done, we apply an adaptive pinning on each spectrum. It's a technique that's used in order to slice each spectrum into different bins, and we try to in such a way that each bin captures a specific substance. Just to reduce this in such a way that each bin captures a specific substance or structure. After that, those bins are the feature inputs for the statistical modeling, which consists of first a feature selection, in which we evaluate the combination of combination of up to 4 features, every possible combination in a logistic regression model. Then we do a five-fold cross-validation of these models to obtain additional parameters in order to select only the best performing models. At the end, when the models are selected, we do a substance identification where we go from the bin notion to the metabolite notion. So here I present the results of the analysis so far. So as you can see here, we are on the training set, we have a performance that is characterized by the AUC, and we have an AUC up to 0.82 in the training for most of the models. And once we selected 35 models to be tested on the test set based on their performance in the training and cross-validation, based on their stability, and also based on their bin quality. And here you can see the results when we apply those selected models on the test set. Here we compare actually the performance on the test set against the performance in the training, and everything that falls in this upper corner is considered as a good performing model. And here we have 8 models that seem to generalize the test set and exhibit a very good performance. So before going into the substance identification step of the bins that constitute these models, we thought if we think of CRPC and no CRPC as 2 forms of aggressiveness in PCA progression, then how would these models actually behave when we apply them on a different use case or a different cohort? And for this, we chose the PCA aggressiveness. Here we have a PCA set with 180 case and 31 control. The cases are patients with a Gleason score that is superior or equal to 7, and controls are considered as indolent PCA with a Gleason score below 6. And as you can see, we just applied those models on this data set without any previous training or anything. And surprisingly, a majority or most of the models actually showed some evidence, showed some potential in discriminating between these two forms of aggressiveness in PCA, which was quite good. And then here I present all the 12 metabolites that are contained in the 8 models that performed well. And we identified 6 metabolites out of these 12 metabolites. And as you can see here, the metabolite 2 and metabolite 5, for example, makes out the majority of the models, meaning that this is like a core model, and whenever we add other combinations, we obtain different performances. The identification step is, however, not as easy, and it's also time-consuming, as we have to look in databases to find which metabolite is it, and if we cannot find the signal in the database, then we will have to carry out additional measurement experiments in order to find the metabolite of interest. So in summary, we showed here that we have a list of models that performed very well in both the training and test set of a CRPC cohort, yielding to a constellation of metabolites associated with CRPC. Then the identification of the aforementioned metabolites is still ongoing, as only 6 have been identified so far, and the same set of metabolites seems also to be associated with aggressive PCA. What we want to do next is that, using the same set of metabolites, we would like to now not only apply them on different use cases, but also train them on these specific use cases. Train this set of metabolites and see whether they have any diagnostic power, like discrimination between healthy and PCA, BPH and PCA, and also train them on an aggressive and indolent PCA set, and see whether they still exhibit any classification results. I also did, during my secondment at GenomeScan, a quick joint analysis of this data that I have obtained so far with transcriptomics data from Mario, but of a different CRPC patient group. And we have found some common metabolomics pathways involved. What I want to do next is to validate these involved pathways by integrating additional data, especially proteomics data, which will be the subject of my next secondment at the Biomedical Research Foundation Academy of Athens. And finally, we would like to have a final validation using assistance biology approach, which will be like a third secondment at Erasmus Medical Center. I would like to thank Dr. Eric Schipper and Dr. Johannes Eisberger for their full support. I would like also to thank Dr. Ines Romer, Mario, and Ingrid for their hospitality during my stay at GenomeScan, and also thank you to all the TransPot members and partners, and thank you for your attention.
Okay, thank you very much, Marouane. That was an excellent talk. So I'll start us off with a question. I was wondering, in selecting the features for your models, why did you select 4 features? How was that assessed as the optimum for your models?
So yeah, as I said, in the feature selection, we evaluate every possible combination up to 4. Why not more than that? Because we don't want to overfit our model. So to avoid overfitting and also for computational powers. So 4 is just a compromise. It's nothing specific.
Okay. Okay. Thank you. So we have a question from Witska. So for progression marker Gleason's greater than 7 versus Gleason's less than or equal to 7, would this be more interesting as Gleason 7s are generally do not progress? Can you make this clinical crucial discrimination?
That is a very good question. The choice of this split point was based on a previous project that was basically specific to the staging of prostate cancer. But what's sure here is that everything below or equal to 6 is definitely indolent, and it's arguable with for patients group with a Gleason score of 7, because it could be a 3+4 or 4+3, which is kind of an intermediate risk. But we decided for now to put them with the aggressive prostate cancer group, just to evaluate whether these markers could have any staging performances.
Very good. Thank you. Can you please give more details related to the tools you use to correlate transcription analysis with metabolites?
Sorry, what was that?
Can you please give more details related to the tools you use to correlate your transcriptional analysis with your metabolites?
Yeah, this was done in joint with Mario, and for that we used mainly metaboanalysis tool that is a public analysis tool that is publicly available on the internet. So we just provide the tool with a set of metabolites that we believe that are differentially expressed in my case between CRPC and non-CRPC, and also differentially expressed genes from data sets provided by Mario, and the tool does an analysis and gives us what are the metabolomic pathways that are enriched given the 2 data sets or given the 2 inputs.
Okay, thank you, Marouane. Another question: What do you hypothesize the difference between aggressive localized prostate cancer with metastatic cohort?
Could you repeat that, please?
What do you hypothesize the difference is between the aggressive localized prostate cancers in comparison to the metastatic cohort?
So I don't actually know, because for my data, for the clinical data I have, even though these patients do not show any signs of clinical or tumor progression, but they could already have a metastasis already diagnosed in there. So I'm not pretty sure whether these cases do not have metastasis already in there, so I cannot judge.
Okay, thank you. So I have another question while we wait for some more questions to come in, but have you tested the prognostic independence of your potential metabolite biomarkers in any multivariate models alongside any common clinical factors for prostate cancer?
No, I didn't do that. We want the model to be only dependent on the biomarkers. I'm pretty sure if we add, for example, I don't know, the age as a clinical covariable, the performance of the model will increase, but it's not significant as because when you do a covariable analysis, you see that the age is already by itself could classify cases and controls. So it's just, you could introduce biases if you introduce other clinical variables.
Yes, thank you. So there haven't been any more questions, so perhaps we can go back to Katie. Thank you, Marouane. Excellent talk.
Thanks.
Thanks, Cathal. Thanks, Marouane. I'm now going to hand over to Craig Robson for closing remarks. Craig is a PI at the University of Newcastle and is also the head of the TransPot Training Subcommittee. Can I hand over to you, Craig?
Sure. Thank you, Katie. So, first of all, a big thank you to everybody for attending the symposium. We've had around about 60 people constantly throughout the time, so I think that's really good. So firstly, a big thank you to all the speakers who contributed towards the symposium, which was entitled The Integrated Prostate Cancer Research Across the European Union. And that's obviously hosted by our TransPot Collaborative. I mean, I'd like to give a big thanks to the two external speakers, Bruno Costa-Silva and Teijo Pellinen, who gave two excellent presentations on extracellular vesicles and multiplexed imaging applied to prostate cancer. Also a thanks to Hing, Leung, who leads our consortium and has been a great leader for this collaborative and for his clear overview of the individual projects in the TransPot consortium. I should say, as Hing sort of indicated early on, TransPot is a European Union-wide consortium of 7 academic institutions and 3 industrial partners. And it's been a fantastic vehicle for delivering research in this area. And as part of the collaborative, we're hosting 11 early stage career researchers who are doing PhDs within the various institutions across the consortium. The major thing to say is that TransPot itself is a research training program, and that's very important that we build on that for the 11 early career researchers who are involved in the collaborative, and just to say that all of these researchers have contributed towards this symposium today, either as speakers during the symposium or as chairs of the individual presentations. I think that the 4 TransPot speakers that we had today provided a good insight to the projects within the TransPot consortium, and I hope that they have actually demonstrated to you the extensive collaborations that occur within the consortium between the various academic and industrial partners, and importantly our multi-omics approach towards the prostate cancer research that we're doing that's employed within this consortium. So we're now in the 3rd year of the TransPot program, and I think we're really seeing now the great progress which is being made by the individual researchers within the projects, but also I think as the PIs in the consortium we can say that the researchers themselves have matured greatly scientifically over this time. So I suppose finally I should say a very special thank you to Katie Wright and Linda Rushworth, who have been very important in helping to organise this symposium, particularly since this meeting was actually supposed to be part of a European Association of Cancer Research special conference, which was to be held in Lisbon last week, and we've obviously had to change that. And I think actually what we have done is actually, I think today we've actually managed to demonstrate that these sort of online symposia conferences that are held can actually work very effectively. And I think just by the fact that so many people today have attended the symposium and also been involved in asking questions, which I think have been a great set of questions for the various speakers, I think it actually has turned out to be very successful. Obviously, we all hope to get back soon to face-to-face meetings and conferences, but I think actually today has been very good, and it's given a clear indication of some of the work that's been going on within the TransPot Collaborative. So that's really all that I have to say. I don't know whether Katie or Hing want to add anything else to finish off the symposium. Hing, Katie, anything?
Yes, thank you very much, Craig. Thank you. I'd just like to extend my thanks as well to everyone who's attended and participated today. We really appreciate your support of the TransPot project. So thank you very much. And that concludes the symposium, unless Hing has anything further to add.
No, nothing. Thank you very much.
Okay. Thank you everyone again for your attendance. Have a nice afternoon. Goodbye.
Bye.